Hello class, my name is Nora Sage, and for my personal project I investigated CRISPR-Cas9 modification of CCR5. So the major obstacle to the elimination of an HIV infection is rapid viral rebound. So what that means is that the latent virus persists for long periods even with HAART therapy. This is due to the constant integration of a proviral DNA into the cellular genome. So when we're seeking a cure for HIV, one method discovered was genetic modification of a CCR5. So currently there are 3 CCR5 manipulations. So there are the transcription activator like effector nuclease, a zinc finger nuclease, or a CRISPR-Cas9. So because CRISPR-Cas9 is known to be more convenient, flexible, and it is much easier as it only requires 20 nucleotides per sequence, and it does not require a complex genome engineering, we will be focusing on this process. So before we dive into the process, I want to first talk about 2 major components of a Cas9. CRISPR-Cas9 technology. So one is the Cas9 endonuclease, which is known to cleave the double-stranded DNA in a sequence-specific manner. The second is a short guide RNA, or gRNA, which is the first 20 nucleotides in which recognizes a target DNA and pairs according to the region adjacent to the 3' end of the target DNA. So in this diagram, you can see here that this is the Cas9, and this red line here is the guide RNA. So now we could talk about the process. So first it begins with initial exposure of HIV as it binds to the cellular membrane and inserts its RNA into the cell. Now, the foreign DNA is then cleaved by a Cas9 nuclease and is captured, and as a result of this, it is then integrated into a CRISPR locus in a form of a spacer. So a spacer, what it does is that it creates a template that forms a CRISPR RNA or CR RNA, and the CR-RNA then binds with a trans-activating CR-RNA to direct the Cas9 nuclease to a complementary DNA. So the specificity of the system is known to depend on the promoter adjacent sequence, or PAM, which is known to be located downstream of the target sequence. So once bound to the Cas9 protein, breaks the CR-RNA with either a HNH or a RUVCL nuclease domain, which acts as scissors, and as a result of this, it will create a double-stranded break, or DSB. Now, DSB is repaired by either a non-homologous joining, which is an NHEJ, or by a homology-directed repair, which is an HDR. But for this, we're going to be discussing the NHEJ. So as shown, the NHEJ is known to combine with the ends of DSB and creates an indel, meaning insertion or deletion mutation, by either disrupting the frame of the gene or by introducing a premature stop codon. So although the improvements using the system are remarkable, there are some limitations to this process. One of the major limitations is what we discussed before, was the NHEJ indel mutation, since it is known to actually prevent the gRNA from binding and DNA from cleaving. So a possible solution to this is actually a CPFL, which is a protein that is known to that cleaves in a more distal region of a target sequence rather than the usual adjacent PAM, which is less critical for gRNA binding. Thank you for listening, and I hope you got something out of this.